So today I'd like to talk about, first, casework validation of genetic calculator mixture interpretation. This is work done in collaboration with Dr. Barry Duesman at the New York State Police, and I'll mention that I work at Cybergenetics, which produces DNA interpretation technology, including the True Allele program, which I'll be mentioning because that's what we're validating. Okay, so there are many ways to look at a likelihood ratio. Before I begin, I want to state a few things. There's human interpretation of DNA and computer interpretation of DNA can be viewed as being essentially the identical process with maybe one change. And I'm going to be describing it that way and pointing out where that one change is by looking at a rule. And moreover, the likelihood ratio can be written down in at least 20 different ways. And instead of using the one that Adele and many other people wrote down, the last talk, I'm going to use the original definition from 1950, which is information gain. They're mathematically equivalent, but I find this one conceptually easier. So the information gain or likelihood ratio is what you learned about an identification hypothesis. Before you look at the data, your hypothesis is, you ask, what are the odds that something happened, like maybe somebody's guilty or contributed to the evidence? Then you look at your DNA data, and you arrive at a posterior state afterwards, and now you have new information. You ask, what are the odds of that hypothesis after I've seen the data? That ratio is the information gain or likelihood ratio. In statistics and other sciences, we take the logarithm, which is the order of magnitude or powers of 10, because then we can add together likelihood ratios. Where do mixtures come from? Well, suppose in blue I have a first contributor, and there's a lot of him, and then in orange I have a second contributor that's supposed to represent their alleles at some locus, and there's less of him. And you add a lot of A to a little bit of B, amplify them, and you get a pattern. That's mixture data with the genotypes of contributors A and B. So if that's independent of your theory, that's just what happens. So let me go through mixture interpretation for both quantitative and qualitative review, computer or human. There are only two steps. You infer a genotype only from the data without ever looking at a suspect, and you have a genotype. Then the second step is you then make a comparison against 1, 10, or if you're using CODIS, 1,000,000 potential suspects and compute a strength of match or a likelihood ratio. So let's look at quantitative mixture interpretation. Every step is the same as for what we all do for human review, except for the rule. Step 1, infer genotype. We consider every possible allele pair. We're required to do that by statistical inference. We then compare the pattern that we form from that pair with the DNA data. So here's an example. Suppose we have a lot of blue, a first contributor, a little bit of orange, the second contributor, and what we're looking at is roughly the shape of the peak pattern. Well, we do a comparison. And the rule is, this replaces a lot of mathematics in this talk, a better fit's more likely it. Not the best rhyme, but it fits in 4 beats and it's good enough to replace equations. So here we see that assuming a major contributor and a minor contributor, and those are the alleles of too big and too little, there's a high likelihood because a better fit's more likely it. Suppose we try to assume that only 3 alleles, maybe the orange shares one with the blue, and try to fit it to that data pattern. It doesn't fit the quantitative data as well. And so it has a lower likelihood. Depending on the problem, it could have a low likelihood or an almost zero likelihood. The result of trying out every possibility, comparing against the data, looking at its likelihood, and then using statistical reasoning like Bayes' theorem, is for every one of 100 possible allele pairs, there's a probability. If the data is very definite, there'll be one answer with a probability of 1. If there's more uncertainty, you'll typically get something bigger and a bunch that are smaller. There's no statement as to what is right. We don't know what the suspect is. We don't know what right means. We only know what the data can tell us. What the data can tell us is a genotype. And a genotype is a probability distribution over allele pairs with a few winners and a lot of losers. We'll see in a minute how human review is identical to this. Step 2 is we match against something afterwards. And we look at the information gain. At the suspect's genotype allele pair, we ask, at that locus, what's the information gain? Again, before we looked at the data, what was the probability of that allele pair? The population distribution. That's what you would believe if you'd studied genetics. You then look at the data, and you get an after or posterior probability of that allele pair based on the data. That ratio is one of the 20 forms of writing down a likelihood ratio. That is the likelihood ratio. And so notice that this is a guaranteed objective procedure because the computer's programmed to have no choice but to do step one without looking at a suspect and only then do step two. So let's take a look at our cases. With New York State, we'd looked at Jamie Belrose will be speaking about the larger scale validation study from which this was drawn in 2 talks. But here we look at where we had a match score. So we're going to look at 2-person mixtures. And here, we don't have a victim reference. So in these 8 cases, the task is to solve for the 2 unknown genotypes, infer them, stop, and then make a comparison and ask about the strength of match. The computer did it on these 8 cases, and the average is 10 to the 13th. Everything's in log units, or about 10 trillion. What's qualitative human review going to do? Well, the same thing. We're going to infer a genotype and then match it. And again, we have to consider all allele pairs, make all comparisons. But the human rule for simplicity is changed. Instead of a better fits, more likely at using the quantitative peaks, it becomes every pair gets equal share. Anything that's included is considered the same, whether it exactly matches the pattern or it doesn't match the pattern. The orange is included either way. And so the result is the likelihoods of, in this case, if there are 4 alleles, there'd be 10 pairs, are all equal. The same inference mechanism, it's all valid. It's just that whatever the true answer might be, or any answer might be, you're not placing bets on what the data is telling you is most likely. You're placing your bets equally. That's the inclusion method. And so now we do the match, but because we tend to lower the probabilities of what we had before, we expect a lower ratio and lower information gain, a lower likelihood ratio, and that's what we see. Human review with CPI is shown in orange on the same 8 cases, and what we see is instead of an average of 10 to the 13th, we have an average of 10 to the 7th, or about 10 million. The average information difference is about 10 to the 6th, or about 1 million to 1. Okay, so same data, identical ways of interpreting data. Your procedure may not be what you do, but the principles are identical. And the only difference is, are you using the quantitative peak heights or not? In both cases, we're looking for 2 unknowns. Now, this is interesting when we start looking at the amount of information relative to mixture weight. We see that on a 50-50 mixture, there's a dip. So you only gain about a factor of 1,000, not maybe 10 billion or so, because you lose the deconvolving information to separate that out with a 50-50. How reproducible is this? Well, with a computer, you can run a statistical program twice, and we see that on duplicate runs, we can calculate a standard deviation, and it's on the order of a few tenths of a likelihood ratio unit, far less than the sample variation that you have in likelihood ratios. So it's quite reproducible. We did the same study on another 8 cases. This time the victim was known in these 2-person mixtures. So the task is different. Infer 1 unknown contributor. If you're doing it manually, use combined likelihood ratio, as it's called, where you use the victim. Here we have more information. Each bar represents, for each case, the log likelihood ratio. And the information that we got is 10 to the 17th, which is like 100 quadrillion or something. So you tend to get more information. Again, in every case in orange, we see the CLR that New York State reported. We get a per case improvement, and the overall difference is 10 to the 4.6, or about 50,000. The 50-50 information loss is not there anymore because you have the extra information of the victim's genotype. The method's reproducible, this time to the order of a standard deviation measure in hundredths of a log likelihood ratio. So here's the summary in numbers and words of the validation study. We used quantitative computer method, better fits, more likely it, relative to qualitative human review. Every pair, every allele pair has equal share. And we look at the improvement. So with 2 unknowns, without using the victim, this becomes CPI, and that's the computer solving for 2. We get 10 trillion, or about log of 13, whereas without using the quantitative data, you get 10 to the 7th, or about 10 million, and that ratio is the improvement, and it's about 1 million. When you do have the victim, you tend to get more information because there's less uncertainty, and the task is to solve for only 1 unknown genotype. There, the numbers get a little bit higher by 100 quadrillion. Human review improves also, but the difference is still on the order of 50,000. What's also interesting is if you compare, as we'll see in the next talk, what happens if you have 1 unknown with the victim, with a mixture, quantitatively versus CPI. There, 17 minus 7 is 10 orders of magnitude difference, or about 10 billion. And that's what you would expect overall when you have these different methods. So in conclusion, the information gain, or likelihood ratio, is a universal DNA metric that can be applied to any way of interpreting mixtures, because implicitly or explicitly, you're inferring a genotype as a probability distribution. Once you have it, you plug it into a likelihood ratio formula and you're done. We looked at the efficacy. We saw the computer always extracted useful information. We looked at the improvement, and we saw that with a victim known, inferring 1 unknown, that there was about a 50,000-fold information gain. And with the 2 unknown relative to inclusion, there's about a 1,000,000-fold information gain. The method is reproducible within tenths or hundredths of a log LR unit. This is objective. People have proposed sequential unmasking, but a computer inherently does parallel unmasking, right? If it doesn't know anything about the other data that you're looking at, victim, it doesn't know about suspects or evidence. It only knows what you're giving it. So when it infers evidence, it only knows evidence. When it infers suspects, it only knows suspects. All this is computed in parallel, and then matched afterwards. It's inherently objective. New York State reported 1 statistic for roughly every 3 items, whereas the computer reported a usable statistic on basically every item. So if you're concerned about reagent costs or time, then you might be getting more results faster using quantitative review. And these results have applicability in scientific studies of different methods, investigation, for searching databases, and for evidentiary applications, as we'll see next. I'd like to thank the staff at Cybergenetics, Jamie Belrose, from NERFI, and the New York State police examiners who pulled out these cases and identified them. There are no endorsements expressed or implied by any organization, and we've written this paper up into a lengthy but interesting manuscript where all the math is in the appendix, and it's mainly examples and descriptions and showing the results. If you'd like a copy of that, please send me an email at perlin@cybergen.com and expressing your interest, and I'll send you a manuscript. Thank you.